Okay, welcome back to Chapter 5 of Biology 1110. We are talking about the biomolecules, and I hope we will be able to get through proteins and possibly DNA in this video. Maybe we'll need to make 2 more. So proteins are our third biomolecule to talk about. We talked about carbohydrates in part 1. We talked about fats in part 2. Proteins are responsible for most of the stuff that your cell is actually doing. Proteins are about 50% of the dry mass of your cells, so most of the biomolecules of your cells are in proteins. Some proteins are enzymes, so enzymes we see right here, they're able to speed up chemical reactions. So we already saw examples of enzymes that help with dehydration, synthesis, or hydrolysis reactions. Proteins do other things. Let's go through some pictures here. Some proteins are contractile.
Contractile proteins, like the motor protein shown here, can move parts of a cell. Other contractile proteins, working together in muscle cells, can move a whole animal.
Okay. Some proteins are defensive proteins.
The immune system makes defensive proteins called antibodies that bind to invaders, such as the virus shown here, and mark the foreign objects for destruction.
Okay. Some proteins are enzymes. We just talked about that.
An enzyme is a protein that changes the rate of a chemical reaction without itself being changed into a different molecule in the process. Enzymes promote and regulate virtually all chemical reactions in cells. An enzyme is a protein that changes the rate of a chemical.
Some proteins are regulatory. They're able to regulate what genes are being expressed.
Gene regulatory proteins bind to DNA in particular locations and control whether or not certain genes will be read. This allows cells to become specialized for different functions and respond to changes in their surroundings.
Okay. We have hormones.
Signal proteins include hormonal proteins that help coordinate an organism's activities by acting as signals between cells. For example, insulin, a hormonal protein secreted by the pancreas, signals in animal cells to take in and use sugar. The hormone receptor is also a protein.
Okay. We have receptors. So, we just talked about insulin binding here. Binding to a receptor, that's also a protein.
Receptor molecules bind to signal molecules and can then emit second messengers which trigger changes inside a cell. Receptors are thus important links in the system of communication among cells. Some signal molecules, such as hormones, are also proteins.
Okay, we have sensory proteins.
Sensory proteins detect environmental changes such as light and respond by emitting or producing signals that call for a response.
Okay, storage proteins.
Storage proteins stockpile building components that cells can use to make other proteins. Storage proteins in seeds provide raw materials used by the developing plant, unless an animal eats them first. Ovalbumin, the main substance in egg white, serves as a storage protein for developing chick embryos.
We have structural proteins.
Structural proteins have many functions. Like tent poles and ropes, they shape cells and anchor cell parts. They may serve as tracks along which cell parts can move. They bind cells together, making organized units such as muscles, ligaments, and the tendons that bind muscles to bones. The silk of spiders and the hair of mammals are also structural proteins.
Transport proteins.
Transport proteins carry molecules from place to place. The example shown here allows certain solute molecules to enter the cell.
Okay, so those are all different things that proteins can do. You need to be very comfortable. If I give you a situation, you can tell me that it is something a protein does or it's not something a protein does. One of the most important things we're going to talk about with proteins are their ability to be enzymes. So enzymes are catalysts. They make a reaction happen, without that enzyme there, without that enzyme there, that reaction would take so long to happen that it's unlikely it would happen at all without the enzyme. Enzymes also can perform functions repeatedly, so they're going to just keep repeating. They're not used up in the reaction. So here you can see this enzyme is breaking apart this molecule into two separate molecules. So you start with the enzyme. At the end, it's broken into two molecules, but the enzyme is still here. So you have some digestive enzymes that help you digest the foods you have. All kinds of enzymes that regulate all types of metabolism without being consumed. So it's available to keep cycling through and doing the reaction again and again. Proteins are made from 20 amino acids. Amino acids are the building block or the monomer of proteins. And a polypeptide means that you have several amino acids joined together. A protein is going to be a biologically active polypeptide. So a polypeptide by itself, because it hasn't yet been folded up, isn't quite active, but a protein is. A protein can be made of one polypeptide or more than one of them. These are your 20 amino acids. If you go on to take more biology classes, you will learn more about them. You'll eventually be able to draw out all of these. But when we're looking at our amino acids, some of them are nonpolar. So here are some amino acids that are nonpolar. So they're going to be hydrophobic. They're going to try to fold into a protein in ways that they don't have to interact with water. These amino acids have polar side chains, so they're hydrophilic. They're happy to interact with water. These are acidic and these are basic, so they're able to happily interact with water. They are going to be hydrophilic as well. These will also turn a protein into an acid, and these will help turn a protein into a base. If you go back and look at all of these amino acids, you see they all have the same kind of structure to them. They have this alpha carbon, which is a central carbon, and it is bonded to an amino group, this NH2, a carboxyl group, the COOH, a hydrogen, and this R group, the side chain. So the R group is what's different in each of your amino acids. So you can see these different R groups. Each type of amino acid has a different R group. Okay, and then amino acids are going to be linked to each other by peptide bonds. A peptide bond is just a covalent bond, but it gets a special name when it is in a protein. It is a peptide bond. A peptide bond connects two amino acids. So you can see here's its NH2, here's its CO, and then the OH here, and the H here left as water to create a peptide bond. Here this OH and H are going to leave as water to create a peptide bond. And so now you're developing a polypeptide chain. Polypeptides can be a few amino acids to several thousand amino acids. Each polypeptide is going to have an amino end, so an amino terminus or N terminus, and then a carboxyl end or a C terminus. Let's watch a video about structure. We're going to talk a bit about protein structure. This is really important for you to understand these different levels. There's also a video, an Amoeba Sisters video, and perhaps a crash course video. If you don't understand protein structure, you need to do whatever it takes to understand protein structure. Whether you are finding more things on YouTube, working with your study group, or coming and talking to me, reading through your textbook, whatever you need to do, make sure you understand this.
Most proteins are folded into a complex globular shape. Each protein molecule consists of one or more chains of amino acid monomers. The amino acids are linked by peptide bonds, so a protein polymer is often called a polypeptide. Because they are so complicated, proteins are usually described in terms of 4 levels of structure.
Okay, so the sequence of amino acids, just what amino acids it has, is going to determine how that protein folds and develops interactions both in small local areas and as a whole protein. And the structure is going to determine how it works. So here's this antibody. is able to bond to this flu virus because it fits. If an antibody didn't fit, it wouldn't work. We see the same thing with all types of proteins. An enzyme has to fit with its substrate to be able to create a catalyze a reaction. So structure of something determines its function. The protein sequence, or the amino acid sequence of a protein is called its primary structure. So here this amino acid sequence is just all these amino acids put together. That's a primary structure of a protein. Hopefully that's pretty straightforward. Then these amino acids are going to start either coiling together or folding into different sheets. These are called an alpha helix or a beta sheet, just in local small areas. This is its secondary structure. Secondary structure is pretty much determined by hydrogen bonding between the different amino acids that are there. Then these coils and sheets are going to start folding together into some three-dimensional structure. That three-dimensional structure is going to be mainly because of the different R groups it has. The hydrophobic R groups are going to turn in, and the hydrophilic R groups are going to turn out. You'll also have some van der Waals interactions between those nonpolar side chains. You might start getting some ionic interactions and some like disulfide bonds happening. And for some proteins, that is as far as their folding gets. Their tertiary structure allows them to be biologically active. Some proteins need to have a bunch of pieces fit together, and so they'll have quaternary structure. And quaternary structure is how each of those polypeptides with their own tertiary structure fit together. Okay, so the primary structure is a sequence of amino acids completely controlled by your DNA. Secondary structure are alpha helices and beta pleated sheets. And these are just going to be how your amino acid backbone interacts with each other. So this is mainly the backbone and not the actual R groups interacting here.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
Okay, so here's a picture of an alpha helix. There's nothing else to get from here, just that's what an alpha helix looks like. And a beta pleated sheet, a computer model of it. That's how it looks folded together. Okay, then tertiary structure, which gives you the overall three-dimensional shape. This is mainly R groups interacting. So we saw it was that backbone that gave it the secondary structure, forming helices and sheets. The R groups will interact to give you your tertiary structure. So this includes hydrogen bonds between different R groups, ionic bonds between different R groups. So here you have ionic bonds. Hydrophobic interactions and van der Waals interactions. So as you have the nonpolar regions turning in, hydrophilic regions turning out, and you can also get disulfide bridges. So here you have two amino acids that both have the sulfur in their side chain, and they're going to form a bond with each other. And that's a covalent bond that really helps hold that tertiary structure.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
Okay, then we have quaternary structure. Quaternary structure is only going to be in proteins that use more than one polypeptide chain. And the way that those polypeptide chains fold together gives you quaternary structure. So here's one protein that has these 4 different units in it. Here's hemoglobin with 4 different units in it to make hemoglobin. Collagen has 3 different units twisted together. So collagen is a fibrous protein. It has these 3 coils together. Hemoglobin is a globular protein, so while collagen is a fiber, hemoglobin is more like a circular shape. It's a globe, and it's got 4 chains together.
Some proteins consist of 2 or more polypeptide chains. The 4th level of protein structure, quaternary structure, results from the combination of 2 or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of 2 kinds of polypeptide chains. 2 of each, a total of 4 chains, make up each hemoglobin molecule.
Okay. Changing just one amino acid in the protein can affect all the rest of that protein's ability to form its shape. So sickle cell disease is a disease that is only this one glutamine changes to this valine, and that change in primary structure will give it a change in secondary and tertiary structures that results in a change in a quaternary structure, so it's not able to form this proper hemoglobin. So it's not able to carry oxygen. It's going to aggregate into a fiber and not carry oxygen through your body. And so you end up with sickle cell disease just because of that one amino acid being changed. So besides primary structure, physical and chemical conditions can affect your structure of your the structure of your proteins. So changing the pH, changing the salt concentration, changing the temperature, and other environmental factors can cause a protein to unravel. So think of like an egg white at room temperature is going to be kind of that viscousy, slimy kind of texture. But if you heat it up to 100 or 200 or 300 degrees, it's going to solidify into a solid egg white. That is because the proteins are denaturing. That temperature is changing the shape of the egg white. When a protein loses its native shape, that is called a denatured protein. And a denatured protein, you can look and see this shape does not have the same shape as this. Because it doesn't have the same shape, it is not going to have the same function. So denatured proteins are inactive. Sometimes proteins are able to fold themselves. Sometimes they need assistance from other proteins. Chaperonins are protein molecules that can help fold other proteins. Misfolded proteins can lead to disease. So if you've heard of Alzheimer's, Parkinson's, mad cow disease, these are all caused by misfolded proteins. Okay, I think at this point I'm going to stop this lecture video and we'll come back for a 4th part to discuss nucleic acids. I will see you in a little bit. Bye.